Our first speaker is Beth Tamburini. That's actually, I think that's easier. No? She received her bachelor's at CU Boulder and came to the campus to get her PhD in molecular biology and finally did a postdoc in immunology here on campus. She's currently an associate professor in the Department of Medicine in the GI division. Her talk, the title of her talk today is Methods for Evaluating Vaccine Antigen Trafficking and Archival.
Great, thanks for coming today. I'm going to talk to you about a project that we have going on in the lab that hopefully you'll hear about it. So in our lab, we study the immune system. So just to briefly go over how immune activation works is to get infected by a virus or bacteria or parasite, so a pathogen, and then you have an innate immune response, and this involves several different cell types like neutrophils, NK cells, macrophages, and these antigen-presenting cells or dendritic cells, which acquire the antigen, so a portion of the virus, bacteria, or parasite, and become activated through pattern recognition receptors, which recognize specific patterns within the pathogen. This causes the dendritic cell to move from the site of infection to the local lymph node or the draining lymph node and initiate the adaptive immune response, which leads to both cellular and humoral immunity and clearance of the pathogen. And this happens in about 7 days. Following that, the antibodies persist for a while, as do memory T cells, and these can be tissue resident or circulating central memory T cells, and these can last for months to years. So the timing of this response is really important for living this. So in our lab, we're interested in a variety of things, but one of the things we're interested in is vaccination. And so there are lots of different types of vaccines, including protein subunit vaccines, which are part of the pathogen that you inject, virus-like particles, which are not real viruses, but just parts of the virus. DNA and RNA-based vaccines, as an example, the COVID vaccine is an RNA-based vaccine. There are non-replicating viral vectors and replicating viral vectors, such as adenoviral vectors, and there are also whole vaccines which are either inactivated or live attenuated. So what I'm going to talk to you about today primarily is this protein subunit vaccine. So this is a protein with an immunologic adjuvant. So this is something that's going to activate those pattern recognition receptors that I told you about on the dendritic cells. And this can be things like double-stranded RNA, CpG DNA, diacyllipoprotein, or phosphorothioate DNA. So what are the factors that promote protective immunity to vaccine antigens? That's what we're interested in. So way back when I was a postdoc, we discovered this process that we termed antigen archiving. And what we found is that following the injection of a vaccine antigen into the skin or into the muscle, the vaccine antigen drains to the local lymph node. And upon entry into the local lymph node, the lymphatic endothelial cells, which line the outside of the lymph node and branch into the center of the lymph node, shown here, acquire these foreign antigens, so these vaccine antigens. And they stay within these lymphatic endothelial cells for a long period of time. And we'll talk about how long they actually stay in some later slides. But this image on the right is showing that these lymphatic endothelial cells, which are shown in red here, are co-localized with the antigen, which is in green. So this is the edge of the lymph node as shown over here. And this over here is a lymphatic endothelial cell that is decorated with these foreign vaccine antigens. And so remember, the lymphatic endothelial cells also line the lymphatics that drain to the local draining lymph nodes. So what we were really interested in is understanding whether or not this acquisition and archival of these foreign protein antigens by these lymphatic endothelial cells were important for promoting any type of immunity. And so what we were interested in was in T cell mediated immunity. And so what we did is we transferred memory CD8 T cells, so just some memory T cells, into mice that either did or did not have archived antigen. And these CD8 T cells were specific to this type of antigen. And then we waited and we challenged these mice with the antigen that the T cells were specific to. And we asked whether or not the presence of the archived antigen was important for enhancing protective immunity. And what we found is that when you have the archived antigen, that you have increased protective immunity and increased number of antigen-specific T cells. And when you don't have the archived antigen, you have fewer. So it seems to be important for enhancing protective immunity. And one of the questions we often got asked was, does the antigen just stay in the tissue site and then drain to the lymph node over time? And is that why you're seeing this increased protective immune response? And so what you can see here is we've injected the ear of the mouse, and in red are the lymphatic capillaries that drain into the local draining lymph node. And at day 1 post-immunization with a fluorescently labeled antigen, you can see there's lots of green, right? So the antigen is there. But by day 14, a time point in which we see the antigen in the lymph node lymphatic endothelial cells, we don't see any antigen in the ear at all, and not with the lymphatic endothelial cells. And this is just quantified over here. So what we wanted to know, because we're interested in how do we increase protective immune responses to vaccines, is how do lymph node lymphatic endothelial cells archive antigens, and can understanding how these lymph node lymphatic endothelial cells archive antigens inform us about how we design vaccines. And so at this point, we didn't really have any idea of how the antigens were taken up by the lymphatic endothelial cells or what the process was for that. And so we used a number of different models in order to try to assess what was actually important for lymphatic endothelial cell antigen archiving. And so these are all of the animal models that we tested that are not required. So using a directed approach, we weren't really able to discern what was required for antigen archiving. And so in order to gain additional information about the cell types and the ways that cells archive antigens, we decided to use a screen. And so what we did is we used RNA sequencing. And at this point, we tried bulk RNA sequencing. And so this was done by a graduate student in the lab, Erin Lucas. And what she did is she immunized mice with this fluorescently labeled antigen, and then she waited for 10 days post-vaccination. And she sorted the cells based on PD-L1 expression. And she did this because at that point, PD-L1 was known to be a marker of different lymphatic endothelial cell types. And you can see that here in green. So the PD-L1 is expressed primarily by the sinus, so the outside of the lymph node lymphatic endothelial cells, where it's not really expressed in the medullary region or in the cortex. And so she sorted the cells, so she isolated the cells from the lymph node, and she sorted them based on lymphatic endothelial cells and PD-L1 expression. And she looked at PD-L1 high antigen negative, PD-L1 high antigen positive, PD-L1 low antigen negative, and PD-L1 low antigen positive, and we processed them for RNA sequencing. And while we found a number of different pathways that were differentially regulated between the PD-L1 high lymphatic endothelial cells and the PD-L1 low lymphatic endothelial cells, such as the PD-L1 high, which are on the outside, are really important for telling cells to migrate to the lymph node, immune cells, and the PD-L1 low lymphatic endothelial cells are really important for division and apoptosis. Sorry. And so this work was followed up by Jonathan Schaefer, who has a paper coming out in the next month talking about this. And so, but I didn't tell you anything about the lymphatic endothelial cells with archived antigen, and that's because we had too few cells to capture the transcriptome by bulk RNA sequencing. And so we took a look back at our data again, and we also had some issues with tracking the antigen in the lymph node. So not only were we unable to sort them because there was such a small amount, we also were unable to detect them for very long. So when we look at fluorescent antigen, which is the image I showed you before, we can detect the fluorescent antigen in the lymph node for about 3.5 weeks. But when we detect the amount of antigen that's there via T-cell transfer assay, so this is an assay where you can detect very small amounts of antigen, the readout is just a little bit indirect. So we can't tell which cells have the antigen, we just know the antigen is there. So the time frame between those was not the same. So we thought, can we maybe improve the detection of antigen over time? And so to do this, we collaborated with Jay Hesselberg's lab in biochemistry, and we created an antigen that was conjugated to a single-stranded piece of DNA. And this DNA has a PCR handle, an antigen barcode, and a capture sequence, as well as a biotin tag. And we wanted to do this because we thought that this would be useful for single cell RNA sequencing. And one of the things we initially did was we injected this with normal single-stranded DNA with phosphodiester bonds. And when we did this, the DNA was degraded likely by endo and exonucleases within the mouse. And so what we did to protect the DNA from degradation is we included phosphorothioate bonds rather than phosphodiester bonds within the DNA sequence. And this protects the DNA from degradation. So to show you this, looking in vitro, these are some dendritic cells. Hopefully what you can appreciate is that this signal here, so this is a cell, the nucleus is in blue, it's outlined with these white dotted lines. And what we've done is we've treated these cells with the OVA psDNA, or the antigen plus the psDNA. And the psDNA is in green, the OVA is in red, so the antigen is in red, and you can see that they're co-localized. So the antigen and the DNA conjugated together enter the cell together. And on the right here, I'm showing that they're degraded at the same rate. So this is looking at double positive cells over here in the top right quadrant. Then you see over time, they go away together. It's not like you retain one signal or the other. So we next asked if we could detect the antigen via the covalently linked DNA barcode using single cell sequencing technology. And the reason we wanted to do this is because we could use much fewer cells for single cell sequencing. And so rather than doing the bulk sequencing, we did single cell RNA sequencing. And at this point, another group had published that there are different types of lymphatic endothelial cells in the lymph node, not just these PD-L1 high and PD-L1 low lymphatic endothelial cells. And they can be distinguished based on their transcriptional signature. So this group had taken lymph nodes, done single cell RNA sequencing, as we wanted to do, and then designated different populations of lymphatic endothelial cells, which are shown on the right. And so the first thing we wanted to do following single cell RNA sequencing was prove to ourselves that the normal cells that pick up antigens, which are myeloid cells and dendritic cells, those cells that acquire antigen and have the pattern recognition receptors, we wanted to make sure we could detect antigen in those cells. So day 2 post-vaccination, we looked at different myeloid cell populations. So shown here in this image, these are different dendritic cell populations, and you can see they're positive for antigen. This is actually looking at antigen amount, so via the DNA barcode. And then when we looked 14 days post-vaccination, we were interested in the lymphatic endothelial cells, and what you can see over here is that the lymphatic endothelial cells, 14 days post-vaccination, have very high amounts of antigen within the different lymphatic endothelial cell populations. But as I said before, the problem we were having was really detecting the antigen in the lymphatic endothelial cells. So we asked, how long does the antigen persist in the lymphatic endothelial cells of the lymph node? And we decided to go out to 6 weeks based on our previous data. And what we found was that at day 2, 2 weeks, 3 weeks, and 6 weeks, we find antigen within the lymphatic endothelial cells, but that it does decline over time. So this is in contrast to dendritic cells, which rapidly decline over time because the antigen becomes degraded by the dendritic cells and processed and presented to the T cells. So here at day 21 and day 42, or 3 weeks and 6 weeks, we see there's no antigen. Whereas at day 14, we really only find it in the migratory CBCs or dendritic cells. And that's because as the dendritic cells came into the lymph node, they acquired it from the lymphatic endothelial cells. So just to diagram what I showed you, after vaccination, we find that the vaccine antigens become acquired and archived by lymphatic endothelial cells in the lymph node. And then over time, the lymphatic endothelial cells lose the antigen and it starts to move towards these outer lymphatic endothelial cells, which are in the subcapsular sinus. And that was based on the different designations of lymphatic endothelial cells based on transcriptome. And then by 3 weeks, there's even less antigen within the lymphatic endothelial cells of the lymph node. But how does this vaccine archived antigen actually impact the immune response? And does antigen release or loss by the lymphatic endothelial cells promote T cell memory responses and enhance protective immune responses? And so this was a project that was taken on by Autumn Doan in the lab. And what she wanted to know is if you get a vaccine, and then you, say, a couple of weeks later, 3 weeks later, get an infection with something completely different, what happens to the antigen that's been archived by the LECs? And based on some other studies we had done, what we would predict is that the LECs actually release the antigen at this time point. And this antigen release could then stimulate any T cells that were made, that were activated during the initial immune response, so the memory T cells. And so the prediction here is that during infection, you'll get migration of the virus in the dendritic cells to the lymph node, and that will result in presentation of the antigen, the viral antigen, and activation of the T cells that are specific to the viral infection. But what we would also predict is that the memory T cells that are specific to the antigen in the vaccine will also proliferate and respond because the antigen is being released by the lymphatic endothelial cells. And so looking 5 days, 14 days, and 21 days post infection, so this would be, we're looking about a month or plus after the initial vaccination, we're looking at vaccine antigen specific T cells. So you can see if you give no infection, there's really no increase in the number of vaccine antigen specific T cells, so they're not responding. But at day 14 and 21, a time point when we would expect the lymph node to be contracting and antigen to be released, you see an expansion in the antigen specific T cells to the vaccine antigen. And so at this point we thought, well, this could just be because the virus is causing the cells to proliferate and it's just non-specific activation of these memory T cells. And so what she did then is she did a number of experiments to show that it was actually the antigen that was being released by the LECs. But this was one really important observation, which was that if you wait 8 weeks to a time point when we no longer see antigen being archived by the LECs, so the antigen is completely gone from the LECs, and then you give the infection, so there's no antigen remaining in the LECs, you wait 2 weeks, and then you challenge the host with the pathogen that contains the vaccine antigen, you see no difference in protective immunity. So this is looking at the amount of bacteria in the foot. But if you do the same thing and you give the vaccine antigen and then wait 3 weeks, give the unrelated infection at a time point which we would expect antigen to be released, and then you wait an entire 7 weeks, so almost the same duration as the above experiment, and then you challenge with the pathogen that contains the vaccine antigen, you see a significant decrease in the amount of bacteria in the foot pad. So the vaccine bacteria. So what we would predict then is that the antigen archiving and antigen release during an unrelated infection is important for boosting the memory T cell population. So what we concluded was that lymphatic endothelial cells archive antigens for about 6 to 8 weeks, and that this can be tracked by single-cell RNA sequencing using a DNA barcode conjugated to the vaccine antigen. I also showed you that memory T cells that are specific to the archived antigens can be boosted during an unrelated infection that improves protective immune responses. And what I hope to have shown is that if we can really understand the mechanisms of LEC antigen archiving, we can improve vaccines by potentially targeting these vaccine antigens to lymphatic endothelial cells. And so with that, I just want to thank the lab. This work has been carried out by Autumn, and TAG has also helped with some of this work, and Robert is also working on this project. And then this was a really strong collaboration with the Hesselberth Lab, and that's it.
Do we have any questions from Dr. Tamburini?
Beth, so as far as you can tell, is the archived antigen an intact antigen?
Yes.
Or is it somehow processed in some way?
It's intact. If it was degraded, we wouldn't be able to see the fluorescence anymore. And how do I explain it? So interestingly, if we do some imaging in a live lymph node, we can visualize the antigen on the lymphatic endothelial cells. And then if we have dendritic cells, live dendritic cells trafficking in, we can see them interacting with the lymphatic endothelial cells. And we can see the transfer of antigen into the dendritic cell. And once it goes into the dendritic cell, the fluorophore, it like dissipates because the antigen is being degraded.
Is the time frame antigen specific?
That's a good question. I don't know. It might be. We think that the time frame has to do with the time frame that an example flu is present, right? So it seems like it's about the amount of time in which a flu season occurs, right? So that the idea is that potentially you could encourage T-cell responses during an active flu season. But we have looked at several other types of antigens. We've looked at HSV, glycoprotein B, RBD, spike protein, trying to think about lots of different antigens, but we've never gone out really far, just because the experiments take forever, with other antigens. I'm not sure.
Do we have any online questions?
Any additional questions? Thank you.